Hi, my name is Coline Gianfrotta. I'm a PhD student at the Université de Paris-Saclay in France, and I will briefly present you a graph-based approach to classify minor motifs in RNA structures according to their structural context. So, RNA must fall into specific three-dimensional structure to fulfill its functions. That's why we need to predict the 3D structures of an RNA molecule in order to determine its function. But some interactions stay currently unpredictable, even though they are essential to the good global folding of the molecule. And the most well-known class of these interactions are the amino-like motifs, that are the most frequent long-range interaction motifs that appear in RNA structures. They have a crucial role in folding and also in cellular mechanisms. So we have here an example in 3D, and here too, and here an example in 2D of amino-like motifs. So those motifs are very difficult to predict for several reasons. Firstly, they lack sequence specificity. We know that there are often A's here, but that's all we know. And secondly, they often bind distant regions of the molecule. And thirdly, they are composed of non-canonical base pairs, represented here by red lines with arrows, according to the Lantis-Westhoff nomenclature. And those base pairs are hard to predict because we have no experimentally determined energy for them. That's why we choose to study in particular this kind of motif, and so the purpose of our work is to find common structural characteristics among minor occurrences in order to firstly establish an exhaustive classification of occurrences. And to do so, we are studying the structural context of occurrences. We call structural context all the canonical and non-canonical bonds that appear around the motif. We have here an example with canonical base pair in double blue lines and here non-canonical base pair. So we have to choose a number of nucleotides around the motif to define our context, and in the study I will present here we chose 3 nucleotides. So now I will talk about our method. We represent structural context by graphs, where vertices represent nucleotides or groups of nucleotides, as we can see here an example, and here too, and the edges represent covalent, canonical, and non-canonical bonds. Grouping nucleotides allows us to bring some flexibility in our representation, and we do this because we are interested in the context of the occurrence and not in each nucleotide precisely. And then we compare those graphs with each other. We have here 2 structural contexts and the corresponding graphs. And to compare them, we set up a similarity measure we called contextual similarity, based on the number of canonical and non-canonical edges in the largest common subgraph we have here and also in orange here. And using this measure, we developed comparison graph and clustering algorithms in order to obtain a first classification. But before that, in order to validate our method, we also measure RMSD between pairs of structural context. We want to know if the 3D structures are consistent with the topology of structural context, because it would mean that the local structural context are important for the formation of the interaction, and so we could use structural context in our method for the prediction of minor motifs. So we have here the results on a dataset of 391 occurrences from the PDB. This is a heat map representing the distribution of RMSD values and contextual similarity values for each pair of occurrences. In each cell, there is the number of pairs of occurrences that have an RMSD value between the bounds indicated in the ordinate axis and the contextual similarity value between the bounds indicated in the x-axis. And we see at first that for lower values of RMSD, we have a good correlation of the 2 metrics, because the better is the RMSD, the better is the contextual similarity. And to be more precise, we choose a threshold in RMSD to determine similar 3D structures, and a threshold in contextual similarity to determine similar structural context topologies. We want to minimize the number of false positives and false negatives, and so we choose the threshold indicated in blue line, so it is 0.75 for the contextual similarity and 2.5 for the RMSD. And there are here the statistics we obtain with this threshold. And so we have a very good precision, but a low recall. But in general, we can say that RMSD and contextual similarity are correlated. And after that, I will talk about the classification we obtained. So we have here the graph of the dataset, where each vertex represents an occurrence and edges connect structural contexts that have a similarity higher than 0.75, so the threshold we talked about earlier. We use this graph to make our classification with the clustering method indicated below. Vertices of same color in the graph represent homologous occurrences, which mean occurrences that belong to homologous molecules and that share the same evolutionary history. And this classification we obtained is consistent with the RMSD in most cases. And in our classification, homologous occurrences are generally grouped in same cluster, and the majority of clusters are composed of homologous occurrences. But we have one main cluster with non-homologous occurrences, circled in red, which is interesting. It is composed of occurrences from different RNA families, and all of these occurrences belong to the same family of A minor motif, called GNRA family. So we have here the 3D alignment of the structural context of these occurrences, and we see that this alignment is very good. There's just one part here that differs among the cluster, so it's pretty good. And so to conclude, we show that RMSD and contextual similarity are correlated in most cases, so it means that the topology of structural context is important for the formation of A minor motifs. We developed a method to automatically extract and classify A minor occurrences, and in our classification, homologous occurrences are generally grouped in same cluster, but also a few non-homologous occurrences share similar structural context and 3D structures. So in future work, we'll find out if this classification can lead to prediction of A minor that are modules impossible to predict for now. And moreover, we'll generalize our method to other motifs difficult to predict too. Thank you for listening.